My name is Sherry Flint-Garcia. I work for the USDA ARS, as mentioned. And today I'll be talking about the genetic consequences of plant domestication, with an emphasis on maize because corn, because that's what I work on. So the outline for today is I'm going to talk a little bit about genetics and breeding and the concepts that we need to have to discuss genetic variation, where genetic variation comes from. I will be talking about domestication, including a number of different species, little vignettes about a number of crops, and then I'll do a little bit more of a detailed analysis of what we know about maize, corn, looking at domestication and diversity levels in maize, looking at the inbred lines that we have in maize, and then looking at teosinte, the wild ancestor. So an overview for the genetics and breeding. We have a number of terms that I'd like to cover up front. Genetics is the study of the heredity and the variation of inherited characteristics. And breeding is the art and science of genetic improvement of plants. And so genetics and breeding are intimately related, but they're very distinct in a lot of different ways. The other terms that we need to address up front are genotype, phenotype, traits, allele, and genetic variation. And these definitions are up here. The genotypes are the genes that you have, the variants of those genes, and what those collectively, how they contribute to you or a corn plant. The phenotype is what you can observe about an individual or a population of individuals. A trait is the characteristic that you are talking about. Tall, short, in the case of maize, having big ears of corn, small ears of corn. Alleles are variants of genes, and we'll talk about alleles a lot. And genetic variation is the naturally occurring differences within a population of individuals within a species. So we're going to have to do a little bit of Genetics 101. So for those of you that are geneticists, I'm sorry, we're going to have some review. For others, I think it's important for us to know about the different terms and how they interact together. And this hopefully will be useful for other presenters in this workshop. So the first thing is all organisms carry chromosomes. And chromosomes are made of long strands of DNA, and the DNA contains genes. Genes are made of deoxyribonucleic acid, DNA. The DNA contain what we call the nucleotides or the base pairs. The base pairs are adenine and thymine, and they pair together, so they are a base pair. Whereas we also have guanine and cytosine, GC, and they pair together. So those are the base pairs, the four nucleotides that make up DNA. Down here in the box is what we call the central dogma of genetics, where we go from DNA, what I just talked about, to RNA and to proteins. And so you can kind of think about these terms as I talk about them on the slide. So DNA is transcribed into RNA, and RNAs are generally single-stranded molecules, basically made up of more or less the same four nucleotides. One of them is altered. And these nucleotides in triplets are called codons, and they correspond to an amino acid. So the RNA is more or less a working copy of the master blueprint of a cell or of an organism. And this mRNA, the mRNA here, with the codons corresponding to the amino acids, make up proteins. And proteins are a class of molecules that include enzymes. And enzymes are working parts of the cell. They function in biochemical pathways. They are the working parts that contribute to your phenotype. And so for an example here, these pictures are all taken from Wikipedia. It's a very nice resource. So here's an enzyme here that has a certain shape to it. And you can see these nice little lock and key kind of cartoons. For example, you may have a sugar saccharide, maybe glucose and fructose. And together they come into the enzyme. There is a change in the protein structure, the way the proteins are folded, and that initiates some kind of a reaction where this enzyme might cleave those two sugars apart, and now you have two simple sugars instead of your disaccharide. That's an example of how an enzyme might work. So these are the concepts we need to think about during all this workshop. So one of the questions is, where does genetic variation come from in the first place? One of the major contributors to genetic variation are mutations. Mutations fall into two broad classes. One can be considered point mutations, and these are basically changes or small deletions of base pairs, those ATs, Gs, and Cs. So here, for example, we have some DNA, and perhaps it was originally a CC strand of DNA. If you have something bad that happens to your DNA, this can happen from UV ultraviolet light, this can happen to chemicals in the environment, etc. One of those base pairs will change, and that's a base pair change. And we call that a single nucleotide polymorphism. Another group of, and it's estimated that in maize, for example, every time a maize plant goes through meiosis, that is making gametes for reproduction, each particular cell can come up with 90 new single nucleotide polymorphisms, or SNPs. So mutations are very common, and many of them don't have any effect. Another type of mutations are chromosomal in nature, where you can increase the copy number of each of the chromosomes, resulting in polyploidy, or you can alter large sections of the DNA, of the chromosomes, where most diploid organisms have 2 copies of each chromosome. One may be completely intact, while the other one, if it was mutated with a chromosomal mutation, has a different part of chromosome attached to it, and so that leads to imbalances. Genetic variation also can be caused by recombination, and again, this is shuffling of genes during the creation of gametes during meiosis. And then another class of agents that cause genotypic variation are transposable elements, or these are called jumping genes, loosely. They were described back very early in the 1900s. When you have a gene structure that has a very particular DNA, RNA, and protein sequence, if a jumping gene lands in the middle of the gene, it can completely alter or disrupt the function of that gene and that protein, that enzyme. And then also, on more of a population genetic scale, intermating between populations can lead to variation. So bringing together 2 large groups of unrelated individuals, having them intermate, can lead to a lot of new variation. So this is just a cartoon of a DNA sequence that I have done in maize and its wild ancestors. We have a number of inbred lines of corn and some teosinte relatives. And if you look at the A, Gs, Ts, and Cs, the DNA sequence of this section of the gene, you can see they're color-coded. And you can see that there are regions where there's an absence of 2 base pairs and a presence of 2 base pairs. And we call that an insertion, deletion, or indel marker. These are very common terms. Another type is what we call a SNP, or a single nucleotide polymorphism, where, for example, just 1 base pair can be changed between one group or another group. So where does phenotypic variation come from? That was all genotype. What changes in your genes? How do we change the appearance of the characteristics of the organism being studied? If you're familiar with the nurture versus nature arguments, we can consider the genotype to be the nature part. It's what makes up your constitution. And this is the reason why fraternal twins look different. There was a very interesting story not too long ago about a woman from an interracial marriage and had children of her own, and her 2 fraternal twins were very, very different looking. And clearly, they're not identical twins, they are fraternal twins. But this is a result of the genotype, the genes. Whereas the nurture part of the argument is in the environment. And when you know, everybody knows some people that are identical twins, but you know the difference between them. And that's a result of the environment that they grew up in. There have been some classic studies in humans where some identical twins were separated at birth, and they turned out to be very different people. Okay, that's the environment. What's more complicated, though, is the interaction between the genes and the environment. A very complex set of interactions that lead to a lot of variation in the phenotype. And so once we have all this phenotypic and genotypic variation, how can those 2 different types of variation change? Usually when we talk about genetics and these kinds of things, we talk about the genes being in equilibrium, Hardy-Weinberg equilibrium. And that just means in a population from one generation to the next, all the genes and the gene frequencies are balanced from one generation to the next. Okay, so that's the expectation for a random population. But there are different things that can happen to that population that violate that Hardy-Weinberg equilibrium. One of them is just genetic drift, and that is due mainly to sampling error. if we took this room of people and we were going to start a new colony of people on a desert island somewhere, and I took 3 people at random, the chances of all the genes in this room going to that island are very small. And so we would have what we call a founding effect to make a new population. So that's just sampling error. Another more important thing is looking at selection, and this applies to both natural selection or artificial selection. And this is the survival of the fittest, the ability of an individual to survive and reproduce and to contribute to the next generation of individuals. And then again, gene flow between populations, bringing populations together, intermating, recombining, that can have changes on the population as a whole. So once we kind of know a little bit about the background of the information we're going to be talking about here, genetic variation, we can classify variation into 2 broad groups. And there's no defining line in between these 2 groups. The first group is what we call qualitative or discrete variation. And these traits are usually involving 1 or a few genes. So in the case of maize, most of my examples will be maize. For example, male sterility can be caused by a single gene difference between the plants. This line being sterile, this one being fertile. Albino seedlings can be a knockout of a single gene. Dwarfism caused by major gene differences, cob color, etc., like that. One trait, one phenotype or the other, on or off, tall or short. It's easy to classify into 2 discrete classes. But quantitative variation results in a continuous distribution of the phenotype, and it usually involves many, many genes and the interactions of these genes with the environment, very complex. For example, seed composition, where the amount of starch, protein, oil, etc. a kernel has is caused by many different genes interacting with the environment. The amount of carotenoids in the kernels, you can clearly see that they're arranged in increasing carotenoid order, but can you tell me the difference between one and the other on an absolute level? Not really. It's very continuous. Rootworm resistance, we also saw a nice picture of this earlier, where we saw a susceptible and a resistant, but there are many, many shades in between, a continuous distribution. And the ultimate quantitative trait is yield, where the estimates are very broad-ranging, but could include thousands of genes, or maybe half the genes in corn, for example. So one of the jobs that I have as a geneticist is to try to link the genotype and the phenotype together, try to understand what genes control the phenotypic variation. And so there's a nice acronym that we use called QTL, the quantitative trait locus. And this is basically a region of the chromosome, if this is a chromosome, where we can say in this area there's a gene or a cluster of genes that controls our traits. It's a very loose definition of this gene structure. Another way that we do the linking of phenotype to genotype is by association mapping. And in this case, we're testing very specific genes for their contribution to a phenotype. And it's a very much more precise way of doing testing. And in this case, at the end of the day, we can say, this gene right here controls our traits. You can see these 2 methods are very different, and I'll talk about them more later. So that was genetics, and now we have breeding. Breeding is very different and very similar to genetics in lots of different ways. It is the act of selecting, either this is in the case of plant breeding is artificial selection, people are doing it, not nature, selecting plants with a desirable trait. And oftentimes this is done without any knowledge of the genetics underlying the trait. That's not always the case. So the overall objectives of plant breeding are to decrease the unfavorable variants or the unfavorable alleles that control the trait. This might be susceptibility to diseases and insects and drought, unadaptedness to different environments, and then simultaneously increasing favorable alleles. Those that are involved with increased yield, resistance to these different stresses, etc. And so the first plant breeders on the scene in different eras, thousands of years ago when a lot of the crops were being domesticated, there are stories out there, and I have alternate variants here. It was likely that women were the ones that were selecting the favorable plants. Thank you for nodding. I'll tell my story later. So in a population of plants, whether they had bigger ears or whatever the case it might have been for domestication of various crops, the women, while the men were off doing the hunting thing, the women were busy doing the early farming. They were the early plant breeders. Back hundreds of years ago, when farmers, for example, were starting to settle the United States, farmers would have their family varieties, their heirloom varieties that they preferred for whatever reason or another. Okay? And then back in 1926, Pioneer Hybrid was the first company to establish for the hybrid corn seed industry. So those are kind of the different phases of first plant breeders. And the idea of plant breeding is you have a population of your crop, and it has a distribution of your traits. Say, for example, this might be yield. There are some individuals that are at the average of population mean, 200, some that are very low performing, and some that are high performing. And by selecting the individuals with the best phenotype, the progeny population will have an increased mean with a different distribution. And so that's the basic idea of breeding, changing from one generation to the next the performance of your population. Okay, that was the background on that. Now we're going to talk about domestication. And this is the adaptation to an intimate relationship between a plant and a human being. And this is kind of a world map here showing the centers of origin for domestication. In North America, several crops were domesticated, including sunflower in the United States, in the eastern United States. In Mexico, there was a large center of origin for corn, beans, and squash, the three sisters, if you're familiar with that story. In South America, the potato, tomato, and cotton were domesticated along the Andes. In Africa, sorghum and pearl millet were domesticated. In Mesopotamia, a lot of the small grains and the lentils and peas. In China, soybean and rice. And in some of the smaller islands, the bananas and the yucca. Okay? So domestication didn't just happen in one place around the world. It happened in many, many places on many, many different species. And roughly speaking, domestication all started approximately 10,000 years ago, with some more species being more recent domesticates, so maybe 3,000 or 4,000 years ago. And so one of the striking things is the comparison of the progenitor species to the domesticated species. And here's just a sample of a number of species with the progenitors on the outside and the domesticates on the inside. And you can see there are striking differences between just the seed size here, the fruit size in some cases. And we'll talk about some of these specifically next. But most plant species during the domestication, comparing to the progenitor species, you can talk about what we call the Domestication Syndrome. And across most crops, there has been a common set of traits that have been targeted during domestication, such that the domesticates usually are more robust. For example, in corn, the progenitor teosinte is a highly branched plant with very weak stems, and the corn plant is nice and strong. There's usually a loss of natural seed dispersal, and we call this non-shattering, such that wild rice, when it starts to mature, the seeds will disperse on their own. It's a weedy characteristic to spread the seed far and wide, such that domesticates don't do that. Rice seeds on domesticates today stay intact for harvest. There's usually a loss of seed dormancy. There's usually a synchrony of flowering, so that if you were the only plant for miles round, you could make your own seed. You don't have to wait for pollen from a neighboring plant or something nearby. Larger fruits or grains, this is a major trait. And you can see this here in tomato, where the wild tomato is very small, and the tomato that we know and love today is very large. And then also apical dominance, and this is more determinate growth. The wild species like wild rice, sorry, that's not rice, that's sunflower, thank you, and for example also in maize, are very bushy plants with lots of different flowers and lots of different fruits, such that sunflower makes only one head nowadays, the domesticates. And there's also been a hypothesized another trait, or set of traits, that the wild ancestors, the progenitor species, are more resistant to different insects and drought and other stresses out there. But that one hasn't been proven yet. So I'm going to go through a little series now of slides on single slide on different crops to show you what the wild and the domesticates look like, and talk about some of the key attributes of the domestication. So for example, barley was one of the first crops that was domesticated, and it was in the Mesopotamia regions about 10,000 years ago. And they meaning not me because I don't work on barley, they hypothesized that one of the first traits was non-shattering, such that the wild ancestor had very loose panicles of seed, and at physiological maturity, the seeds would disperse. Whereas the modern cultivars today, either they're 2 rows or 6 rows, the panicles are very compact and the seeds are held on until harvest. And the domestication for barley is such that they feel that there were possibly 2 different domestication events, or they're somehow related, but the blue variety, or the blue domesticates here, contributed mostly to the European and American varieties that are used today, whereas the red varieties that were farther east contributed more to the Asian and Far Eastern cultivars. Now I know all of you are looking at one thing on the slide, and that's the beer. We'll have to wait on that till later. But the wonderful thing about barley is that it has a lot of beautiful diversity within the species. You can see differences in the panicle shape and the awns and the colors. Wheat, likewise, was a very early domesticate, and it has a very complex history due to the polyploidy, that is the chromosome set doublings that have happened during its ancestry, and then all the more subsequent market classes that have been developed for wheat, such that the approximately, I believe, the estimate was about 500,000 years ago, these 2 single diploid species came together to make a tetraploid species, and then the tetraploid species was mated to another diploid species here to make the hexaploid species. And each of these has a different market class today. Einkorn wheat is supposedly supposed to be healthier for you because it has less protein and causes less of the reaction to the glutens and the different proteins, thoracic disease, I guess. And then the tetraploid wheats are the emmer wheats and the durum wheats. Durum wheats make all the pasta that we love today. And then, of course, the hexaploid wheat is what we know as the bread wheat today. Rice also has a very interesting domestication history. You can see that the wild rice has a very loose panicle, and the domesticate is really nice and quite nice and tight. If you look at all the different rice varieties that are out there today, they can be loosely grouped into 5 groups: the aromatics, the Ausch, I'm not able to see this from this angle, the indicas, and the japonicas, both the temperate and the tropicals. And one of the current debates going on now is whether there was a single or multiple domestication events that happened for rice. The prevailing theory, until not too long ago, was that there were 2 independent domestication events, one in China and one in India, leading to the japonica and the indica broad groups. But more recent work is hypothesizing that there was a single domestication event in China, and then the domesticate, the early domesticate, moved into India and replaced some more of the wild relatives that were being slowly domesticated there. Soybean, a major crop in the United States, also has a complex history because of the breeding history. Glycine soja is the wild relative. It's a very interesting plant. It vines. It has very small flowers. The seed pod shatters, spreading the seeds. It was domesticated into the Asian land races about 3,000 to 5,000 years ago. But then when the colonization of the United States happened and they started to introduce varieties, land races into the United States, approximately 86% of the variation in modern cultivars comes down to 17 founders from the land races. So there have been these continual bottlenecks during domestication and plant breeding to make soybean cultivars that we have today. And the traits that were targeted during domestication were determinancy, getting rid of that viney characteristic on the wild ancestor, increasing the seed size, changes in flowering time and photoperiod sensitivity, and then changing the seed coat color, and then the non-shattering pod. Sorghum was domesticated about 6,000 years ago in Ethiopia. They hypothesized that the initial domestication events were targeting the non-shattering seeds and the compact panicles. And sorghum tolerates a number of conditions that are important today to study drought tolerance. They tolerate arid conditions. And again, commodity groups are kind of driving the different subgroups, the subpopulations of sorghum. There's a group of sorghum lines that are used for forage, some that are grain sorghum varieties, and then also sweet sorghum. And tomato is a very good example for genetic systems for investigating domestication. You can see that the wild ancestor has very small berries on it, whereas the tomatoes that we know today are very large with their fruit size. There's a lot of beautiful diversity that is not just residing in germplasm banks, but is now being used by gardeners at home today. The wonderful sizes and shapes and colors of the fruits. The exact details of tomato domestication are a little bit uncertain. There's currently debate whether it was from Peru and moved into Mexico or if the domestication happened in Mexico. But they are quite sure that one of the early domesticates were these little tiny cherry tomatoes related to the wild ancestor. And then it was brought to Europe by either the conquistadors or possibly Columbus himself. But the earliest record is 1544 in Italy. And again, this is a really nice genetic system to study domestication traits and variation because of the huge fruit size difference. In common bean, a couple of these slides were provided by other speakers in this workshop, so I need to acknowledge them. Common bean was domesticated from the wild populations. The wild beans grow all over South America and Mexico and Mesoamerica. And it has hypothesized that the earliest wild beans were from Ecuador, northern Peru. And they were grouped into two different groups. And as they spread, these wild beans became adapted to different environments. And the wild Mesoamericans, wild beans resulted in a number of cultivars that are small-seeded, relatively small-seeded varieties today, whereas the wild Andean ones give rise to a number of larger-sized seeds, and those can be shown here. Another beautiful example of the wonderful diversity that's out there. Again, sunflower is a little bit more of a recent domesticate. This was domesticated in the eastern United States. The graph here with the United States shows the range of the wild sunflower shown in blue, and then the cultivated sunflower shown in red. And you can see these regions overlap, and it's the gene flow between the wild and the domesticates that still continues to give us diversity today. And you can see that there are a number of these traits involved in the Domestication Syndrome that contribute to differences between the wild and the domesticates. Okay, and finally we arrive at the most important crop in the world, and my favorite, maize. And so my question is, what can we learn from using all this information about genetic diversity and domestication to apply this information to breeding and genetics and genomics? Okay, maize is an important crop. I think we all know that. It's one of the major crops in the United States, of course, and around the world, providing food and feed for humans and animals all over the world. So I'm going to give an introduction to maize domestication and maize diversity. I will be talking about some work that I've worked on, done with maize inbreds using the nested association mapping population, and then also some work I'm doing with the wild ancestor Teosinte. And here you can see just a few pictures I've taken over the years showing just amazing diversity in corn. This is a population I've made. And then here's some Teosinte growing next to corn. So when I say the we here, when we're talking about the maize part of the project, the we represents a lot of people that work together on the Maize Diversity Project. This project is an NSF-funded project that is led by Ed Buckler up at Cornell in New York, right here. All the people who are in green boxes are USDA ARS employees who work on the project. It's important to point out that ARS and NSF have worked jointly on this project because a lot of these projects are very large in scale, and having a small group, it's just not feasible to do these large-scale studies with a small group and without all the extra support from USDA. There's a population geneticist in California, Jeffrey Ross Ibarra. John Dobley is very famous for his work on teosinte and domestication. He's in Madison, Wisconsin. The Cornell group works on everything from population genetics to a lot of the genotyping methods that I'll talk about. Jim Holland in North Carolina is a breeder and geneticist. Mike McMullen and I are in Missouri. And then Doreen Ware is instrumental in the Maize Genome project in annotation of the genes. Okay, so it turns out that, again, if we look at all the people in the room, we think that we're a pretty diverse crowd, okay? When you look at the molecular diversity in population, people are very different, but they have a certain amount of genetic diversity among them. And when you compare people, human beings, to chimpanzees, that diversity is much larger. So I want you to pay attention to this number, 1.34%, okay? It turns out that corn is much more diverse than the difference between people and chimpanzees, okay? So take any one of you and compare yourself to a chimpanzee, and then take two inbred lines, not wild ancestors or anything like that, just two inbred lines from around the world, and they're more different than you and a chimpanzee. Okay? So maize is just incredibly diverse. So maize domestication, maize domestication exhibits the classic domestication syndrome. The wild ancestor is teosinte. The subspecies is Parviglumis. It's a highly branched plant that has hundreds of little tiny ears, and the ears are not really ears. They are just seeds that are stacked on top of one another, and so that when the plant starts to dry down, the connection between the seeds becomes loose and the seed shatters and spreads. Again, a weedy trait. Not only that, but the seed itself is very small and enclosed in a really hard fruit case. It's what they call a stony fruit case. And the only way that I open them up on a regular basis is with fingernail clippers. And I don't know how they did domestication without fingernail clippers 9,000 years ago. But 9,000 years ago, somehow, I wish I could go back in time and see how this happened, people were interacting with teosinte in a different way, and then they noticed off to the side, or some part of the population was different than the rest of the teosinte. And they kept it off to the side, and they allowed that population to change over 9,000 years to make the corn that we know today. The beautiful corn here usually is a single corn stalk with one or two ears, and the seeds are naked on the cob. There's no hard fruit case around it. And then all through that process of domestication, there's been artificial selection for various traits. This artificial selection was done by people. And as the early domesticated corn spread from Mexico north and south across the Americas, the corn experienced different environments. A very hot, dry plateau, a very humid and disease-ridden area of Mexico, for example. And every time that this corn adapted to a different environment, we have what we call a land race. The land races are very diverse, open-pollinated populations of corn that are related somehow and are adapted to a very unique environment. And it turns out that if you compare the 700 or so land races from around the world, there's actually more diversity within a land race than sometimes between land races. So land races are very, very diverse populations of corn. And we'll talk about all this as we go. So the Maize Genome sequence was announced a few years ago. Maize has 10 chromosomes. It's an ancient tetraploid. So a long time ago in a land far, far away, there were 5 chromosomes, and each of the chromosomes doubled. And so now we have 10 chromosomes today. And that domestication event, or the duplication event, happened about 12 million years ago. B73 is the reference genome line, reference inbred line for the Genome Project. And it's predicted based on the sequence alone that there are approximately 32,000 genes in corn. And in reality, it's probably closer to 40,000, perhaps 50,000. The Genome Project is not far enough along to exactly give us our final number yet. And through a number of studies, I think I mentioned one of them in the next slide, nearly 85% of the B73 genome, sorry, that's unrelated. Nearly 85% of the B73 genome is annotated as transposable elements. These are those, and retrotransposable elements. These are those jumping genes that a long time ago, millions of years ago, a jumping gene landed in between genes, and the more jumping genes landed in between it. So there's a lot of the genome that's junk DNA. And the definition of junk DNA is changing every day now. Some of that junk DNA is actually very important. This is the point I was trying to make before. B73 actually only includes about 70% of the low copy genes that are found in all of maize. Okay, so just because you have one line that's been genotyped, genome sequenced, doesn't mean you know everything about corn. Corn is so diverse, we just don't have the answers yet. So looking at the diversity on a genome-wide scale, the Maize Diversity Project a few years ago did some large-scale genotyping, sequencing in 27 inbred lines. These are the NAM founders that I'll talk about later. And in looking at just the gene-rich regions, we found 1.6 million SNPs, or indels, among these 27 lines. Okay, remind you that the difference between a base pair difference here is a SNP, and the insertions and deletions are indel markers. So among 27 lines, we have 1.6 million SNPs. These are not SNPs that anybody made intentionally. These are just naturally occurring SNPs. Just recently, within the last couple months, the Maize HapMap genome project came up with, from our project, came up with the whole genome HapMap. And in this case, we took 103 lines, including the same 27 lines, plus some other inbreds, some land races, those are the intermediates between domestication and modern inbreds, and then 19 wild relatives. We found 55 million SNPs, okay? So all this diversity is naturally occurring out there. When you talk about maize and maize as wild relatives, 55 million, that's not all of it. Every time we turn around, we find more SNPs, okay? And then, so that's all at the molecular level. If we look at more of a genome scale, more of a chromosomal level, based on the relationships with species back in time to evolution and through evolutionary time, you would expect that related species, such as maize and rice, would have what we call synteny, or the same gene order for its chromosomes. So here you can see chromosome 1, bottom part of chromosome 1 in maize, and you can see chromosome 3 in rice. And all these little tiny green lines that connect the two chromosomes, those are genes that are in approximately the same order between rice and corn. So if rice and corn have that much gene order going back from millions of years of divergence, we would certainly expect that within maize that the same two kinds of corn would have the same gene order. But that's not true. It turns out that we see a lot of what we call non-collinearity in maize. And so blowing up part of a slide, or of a figure from a publication back in 2002, this group sequenced between two different lines, a line named after Barbara McClintock, who discovered jumping genes, and B73. The orange and yellow regions are genes. All these blue and red and green areas, those are transposable elements and retrotransposons, again with genes. We can see that by comparing these two lines, there's a huge amount of junk DNA that is present in one line and not in the other. Okay, so maybe we don't care about junk DNA. The problem is that this also applies to genes too. When we compare these same two lines, there are actually genes that are present in one line that are absent in the other. So it's not just a matter of having SNPs, it's a matter of having the gene or not, and that's what we call presence-absence variation. And there was another study that was done recently looking at, again, comparing two lines, MO17 from Missouri and the McClintock line, comparing the genes, which are black triangled in this case. And you can see that there are regions that are present and absent in one or the other, and they're generally associated with certain classes of transposable elements. These jumping genes not only can land in the middle of your gene and turn your gene off or change your gene, they can actually pick up that gene and either delete it or move it someplace in the genome. And these are not free kinds of corn. These are just corn, natural varieties that are out there. So moving more away from the diversity and into domestication, back in 1979, 1980, George Beadle, famous geneticist, did some early studies looking at the genes that control domestication or that were responsible for domestication. And he proposed that there are 5 major genes that can distinguish corn from teosinte. And there's a region on chromosome 1 that I'll talk about next, I believe, and a region on chromosome 4 as well, that control differences in the plant and that seed morphology. So back in 1991, and he did that all based on just crossing corn and teosinte and looking at the progeny. Back in 1991, John Dobley did a QTL analysis, looking at specifically what parts of the chromosomes come from teosinte and corn to cause the differences, and found exactly 5 QTLs for those large-scale differences. This was very convenient at the time. So what are these domestication genes that were early proposed? One of them is called teosinte branched, and it causes differences in the branching structure between teosinte and corn. And also, normally where an ear would come off of a corn plant, there's now a long branch that's just like another corn stalk with ears along it. That's the highly structured plant of teosinte. And it turns out that the difference between teosinte and corn is caused by a transposon insertion into a transcription factor, a major key switch gene that controls plant architecture. Another gene called teosinte gloom architecture controls that hard fruit case around the seed that's absent in corn and present in teosinte. And they worked down the genetics to a transcriptional regulator, again, that they don't know the exact mechanism yet. They think it's just a SNP in the gene that causes whether that seed coat is big enough to cover the seed or not. Okay, so these are very large effect QTL, regions that control a gene, a trait. So now that we have genomics as a tool, we have moved from studying one gene at a time to looking at the whole genome. So back in 2005, there was a study where they sampled 700 or so genes across 14 inbred lines. And by doing a number of different statistical analyses, they found that 2 to 4% of the genes in maize were selected during domestication or plant breeding. Okay, this, if you interpolate this to 50,000 genes in maize, this is over 1,000 genes that are responsible for differences between teosinte and maize. That's very different from that original number of 5. Okay, and then recently, now in 2012, the group has come up with a whole genome analysis of the HapMap version 2. I talked about this before, looking at these 55 million SNPs across maize and land races and teosintes, and they've identified a number of key findings here. The idea is that land races retain 83% of the diversity of teosinte, with maize not very much lower than the land races. The land races are in red, the inbreds are in blue, and the teosinte is in green. They have, by doing a number of different statistical analyses, found that there are stronger, the selection was stronger during domestication. These XPCLR scores are on the order of 150 for domestication, and much weaker on the order of 30 or so for plant breeding. And again, coming to the conclusion, there are approximately 1,000 genes that were targeted for selection during domestication and plant breeding, again, causing differences between teosinte and maize. So the question is, how can we use all of this information to study corn, to use corn in a way that's beneficial? So this is a cartoon diagram of everything I've said up to this point about maize. So if we look at the different germplasm pools, the teosintes, so land races and the inbred lines, for the vast majority of the genes, 98% of the genes, if we look at the alleles, the variants of genes, of a random gene, you can see that they're colored here in a cartoon fashion. And during domestication or any kind of demographic change in the population, there is some loss of diversity due to a bottleneck. Perhaps we've lost the purple allele. And then during plant breeding, maybe we lost that red allele. But the idea is that there's a lot of wonderful variation still in corn today that we can use in breeding in genetic studies. On the other hand, when you talk about a domestication gene or a plant breeding or improvement gene, the situation is very different. When you talk about domestication, that bottleneck during domestication was very severe, so that only one allele or a small number of alleles made it through that bottleneck. And so the idea is that, of course, we don't want that hard seed coat gene back in corn, but maybe there are a number of other genes that were lost during domestication that we might find important today. Likewise, a similar thing happened during plant breeding, forcing corn to grow in straight lines under high density using fertilizers and combines, etc. And my research project involves using all these different tools, these populations, using genomics and genetics and plant breeding to try to understand all of this and to utilize this information. Okay, so I have 15 minutes left, so I think I'm going to zip through this, and hopefully if there's some interest, we can talk about this later. So the first thing I'll talk about is the research I've been doing with inbred lines. This constitutes what we call the nested association mapping population, or NAM. So traditionally, when you do QTL analysis, I talked about this very early on, we do a genome scan across the genome to find regions that control the traits. And typically, two parents are crossed, and a population segregating is developed from that. We use markers to genotype. We phenotype the plant for our trait of interest. We do some statistics such that along the chromosome, we have some region that seems to be important for our trait. And the problem is that region may still contain 1,000 genes. Association analysis, on the other hand, is very much more precise. We're looking at a specific gene and asking a question about that gene. So for example, if we were interested in plant height, and we took 300 kinds of corn and measured them for plant height and arranged them in increasing order, and we think that gene X is involved in plant height, we would sequence that gene and find the SNPs, or the indels, that are different between different lines. And if you look closely, you'll see that one particular SNP segregates. All the short kinds of corn have an A, and all the tall kinds of corn have a G. And that's, in essence, what association mapping is about, but it's much more complex with statistics. So combining the two different methods together into what we call NAM, we can combine the high power of one method with the high resolution of another method to come up with a really unique tool. And so one of my first jobs as a postdoc with Ed Buckler was to make things bigger. He had 100 lines that he was studying, and he said it wasn't big enough. So I went into Major Goodman's cold room and took out 200 more inbred lines of corn. We had them genotyped, and now we can see the relationships among these 300 inbred lines. Each of these tiny little lines with a little tiny text at the end signifies a single inbred line. And you can see that B73, the line for the Genome Project, is here. It's in a group called the stiff stocks. Mo17 is a line that's in the non-stiff stocks. They're in blue. We have sweet corn lines, popcorn lines. They are very distinct germplasm groups because of their breeding histories. And then we have all the tropical and subtropical lines in green. So when we think about diversity, 300 lines for one application is a good size, but we wanted a core set of 25 lines that were maximally diverse. So based only on markers, we chose the 25 founders of NAM, and they're shown here in red. They were all crossed. The 25 diverse lines were crossed by B73, and from each of these crosses, then a population was made. And so in looking at, in this dimension, we can utilize the strengths of linkage mapping while using the ancestral recombination that's gone over the breeding histories of the lines, we can do association mapping. And when we grow these corns up, 1 minute left, okay, plus 10 minutes for questions. When we grow this up, it takes close to 6 or 7 acres to evaluate these things. These are 3 fields of corn. And we can't possibly go out and measure 6,000 rows of corn at one time very efficiently by hand. So we've developed a system called Corn Tracker where we put barcoded plant tags on the plants, and we've used barcode readers to go out and use barcode plant tools, integrate, and input all this data directly into a computer. A postdoc on my project is working on kernel composition, clearly a trait that's very important to this group. I will focus on starch, protein, oil here. And we identified QTL along the 10 chromosomes of corn. And this particular green peak here is a QTL. Again, we can say at the end of the day, somewhere in this region is a gene that controls oil. We used genome-wide association mapping using the Maize HapMap version 1 of 1.6 million SNPs. Okay. To do some statistical analysis. And we are getting much closer to saying that this particular gene is involved in oil content. We think we know what the gene is. It's a gene involved in oil synthesis, GWAS. Pioneer cloned this gene a number of years ago and found that the high allele contains an extra amino acid in the protein. And we found that our top 4 alleles have that same extra gene, phenylalanine insertion in the protein. And we can start to look at haplotypes of these SNPs across the gene that explain quite well our allelic series, such that the bottom alleles here are all non-B73 SNPs, and a number of haplotypes, or a number of lines have another haplotype where part of the gene is from B73 and part not. But we have an interesting allele that we don't understand yet. Okay, we'll skip that. Very, very, very quickly go through teosinte and using the wild ancestor. In an effort to try to get at some of these domestication genes and to ask questions about their value today, I have taken B73, the line that's being sequenced, crossed it to 10 different teosintes, and made back-crossed to make near isogenic line populations that look very much like corn. But when we look at the different chromosomes, we can see that most of the genome is like corn, but there's small regions of teosinte in them. And so now, instead of comparing this big weedy plant that looks nothing like corn, we can ask questions about very specific regions. So there are 1,000 selected genes. What do these genes do, and are they important to us today? What traits are they associated with? Clearly, certain genes control the branching habit of corn. Is that possibly valuable to us today? I have a postdoc working on the project who is trying to find the gene that one of the genes that controls how many kernel rows go around the ear. B73 has approximately 18 kernel rows. Some of my teosinte lines have closer to 8 or 10. And we have a region on chromosome 2 that we're very interested in that really has a very dramatic effect. Likewise, for 50 kernel weight, seed size is also a target of domestication. And then to reintroduce some variation into corn. In the starch pathway, there were a number of genes that were targeted by domestication. Maybe by putting more teosinte in, getting different allelic variants in the population, we can make bigger, and better, and safer, more healthier, different kinds of starches. And I'm interested in many different kernel traits, which I won't go into. And here's my crazy idea. Okay, so back 9,000 years ago, there was this teosinte plant. People discovered it. Like I said, people hypothesized it was a woman who did the early domestication, the early breeding. But when I get really busy at home, I usually have my son go out and do something for me. Hey, Eric, can you go pick those potatoes in the garden for me? Oh, Mom. So every time I touch this teosinte plant, it falls apart. Every time, it's just so many little seeds, and kids are lazy. Kids want to take shortcuts. And so probably kids were the ones who were doing the early transformation of corn. But here we are, 2012, soon to be 2013. We don't rely on our kids or ourselves to go out and harvest corn anymore. We have combines, and the combines don't care about how many ears the corn plant has. So what if we could reintroduce teosinte and start selecting for high yield, and it didn't matter what the corn plant looked like? Would we end up with something like teosinte or something like the corn that we have today? And I have some populations that are high proportions of teosinte, and we're starting some experiments to address this question. I'd like to acknowledge ARS for, of course, my salary and research support, the Maize Diversity Project for all the intellectual contributions, and then also some corporate people who have done generated some materials and done some analysis for me. With that, I'll take some questions. And I'll leave a quote here that you can read if you want to from Darwin about artificial selection. Thank you. The question is about this presence and absence from one line of corn to the next. How much is there? Is it completely gone or just moved? The early estimates are that up to, and it depends on whether you're talking about all of corn or just specific comparisons, between any two kinds of corn, there may be 10 to 15% of the genes that are in one line that are not in another. Okay? And across all of maize inbreds, there are probably 30% of the genes that are in different places. And whether they have implications on the traits, the initial analyses seem to be that the presence-absence variations are very important. Okay, so the question is, if we're talking about 10% across two inbred lines, that would approximately be 4 1,000 genes that are present or absent comparing two lines? Yes, that's the answer. And it may not be exactly that they're present or absent, but they could have been moved by these transposable elements to an entirely different part of the genome and be under the control of some other gene that we don't know about. And these happen all the time. The question is being about mutagenesis. So in some species where there is not much diversity, people can actually use mutagenesis to create variation. It should have been on one of my earlier slides, but it wasn't. Yes, many species are very low variation, and they do intentionally use chemical and radiation, different kinds of mutagens to create variation that they can then select on for plant breeding, plant genetics. How common it is, it was more common back in the 1970s and 1980s. Certain species like maize have enough diversity that we don't need to do that ourselves. So